I see how it is, Sam. You just wanted it all to yourself, didn't you?
No, I don't think so.
Okay, 1 minute to starting. Just while waiting for one of the colleagues to come back and then we can start. Just wait for to come back. Yeah.
Oh, sorry.
Good. Okay, morning colleagues, morning everyone. Those who are joining us here at SAIAB in the room and colleagues that are joining us online. Welcome to this very important session. Today we have a high-powered delegation from Stellenbosch University, from the African Microbiome Group that is based in the Department of Microbiology and School for Data Science and Computational Thinking. The group is led by Professor Thulani Makhalanyane, and it currently has 20 members that includes postdocs and PhD and MSc students. Their research focus is on understanding the ecology and evolution of microbial communities in different ecosystems, and there's also a growing interest in human health, which is very, very critical, putting that link between the work that we do and how it directly impacts society and humans. He has co-authored several papers. I counted more than 150 papers, and some of them are in very high impact and leading multidisciplinary journals such as Nature, Science Advances, and many others. He serves on several editorial boards and is currently the chief editor of the ISME journal and senior editor of the M Systems. He also serves on national and international panels representing South Africa in bilateral discussions. In this talk, Thulani and the team, they will provide an overview of their research. Thulani is joined by Dr. Nello Gregory, Dr. Nontsikelelo Hlongwa, Dr. Sam Liarang, Dr. Zama Mahlobo, and Mr. Maiwongwe Buteles, who is soon to be Dr. Buteles, I'm sure. He has just recently submitted his PhD thesis for examination, so we are rooting for you, Mike. Over to you, Thulani. Thank you.
So thanks a lot, Albert, for the introduction. As you were saying, some of the papers are published in good journals. I want to just confirm the others are also okay. they're not that bad. So, well, thanks for the opportunity for inviting us to come and speak. The purpose of this trip is to think about how we can start to work together with people here. We've recently moved our group, recently being 2 years ago, to Stellenbosch University. And with that, we're trying to think about how we can start working, particularly in coastal ecosystems. And with this talk, I'm just going to give the introduction. We've got, the so-called high-powered delegation that are going to give the bulk of the talk and talk you through what we have in mind in terms of the vision of what we'd like to do with our research. So, I'm a strong believer that African microbiomes hold the key to the future research on the continent. And we've recently started to think about what we need in order to promote African microbiome research. We've co-authored a paper a couple of years ago where we've started to think about how there is a strong knowledge deficit related to African microbiomes and the type of things that we need to start doing in order to promote research that specifically focuses on African microbiomes. Part of this, we've started to think about how we can coordinate ourselves as African researchers, focus on research on the microbiome. And part of this coordination thinks about how we can set up, for example, continental hubs that specifically focus on African microbiome research. And part of this will include how the type of policy interventions that we'll need to develop that can start advocating for research that focuses on innovation specifically, and also how to increase research capacity that's focused on African microbiomes. A strong part of this, and part of the discussions that we had in the morning with Gwyneth and others, is how we can start to think about the type of infrastructure that we need in order to promote the sequencing efforts that we will need in order to get to the target that we've set for ourselves quite randomly, and that target is to sequence 10 million metagenomic samples in the next 10 years. With that, you need to have dedicated research hubs that specifically promote the sequencing throughout the continent. And we're not keen on having facilities that have sequencers everywhere, but none of them being used. And what we'd like to see happen is dedicated research sequencing facilities, maybe at central points throughout the continent, that specifically coordinate the sequencing of samples throughout the continent. And one of the things that we've started to advocate for is that we work quite a bit with researchers abroad, and we've started to think about how these partnerships often are intellectual projects that are conceptualized overseas, and African participants are merely just the implementing partners where we provide samples. We want that to change, and specifically change within the context of African microbiomes. And we've written a piece that starts to think about how we can kind of implement a framework which would advocate for these research efforts being equitably led from the continent. So our research focuses on a variety of different aspects. I use the word focus very loosely here, but the underpinning thread is that we focus on understanding the ecology and evolution of microbial communities across a variety of different ecosystems. We've got research that focuses on microbial diversity, understanding evolutionary adaptation, and also understanding microbial interactions across different systems. We use the One Health framework as a way of answering ecological questions across these different systems. And as Albert has mentioned, we've established a group where we look, for example, at how childhood stunting is directly influenced by the microbiome, and how we can alleviate stunting specifically in children within the Western Cape. We've got other projects, for example, that focus on human health and also environmental microbiomes, which I won't talk about in great detail. So our group has different focus areas, which I also won't talk about, but the research that everyone will talk about here really touches these 4 different areas. So we're trying to look at the dynamics of African microbiomes, to understand the impact of climate change effects on African microbiomes, to look at how we can set up different niche environmental monitoring sites throughout the continent in order to establish programs that have a strong surveillance efforts throughout the continent. We're also interested quite a bit in microbiomes and specifically the relationship with human health. So these are currently the projects that we have in the lab. We've got a project where we're specifically interested in looking at acidobacterial genetics, phylogeny, and also functional mechanisms. This is an NSF NRF funded program. We've got a project that we've just about to finish where we're trying to use microbiomes as sentinels of change in coastal and marine ecosystems. We've got a new program that is really an offshoot from the fundamental research that we've done. And in this program, we're trying to use machine learning approaches to try and leapfrog the discovery of microbial byproducts in different South African ecosystems. We've also got a program where we're looking at understanding the persistence and transmission of antimicrobial resistance genes in African wastewater and freshwater ecosystems. Sam is leading that project, and I think we've got about 11 partners throughout the continent who are participating as part of these efforts. And the idea is that within the next few years, hopefully, we'll have a gene and genome catalog that fully describes different wastewater systems across the continent. So this is the group, the African Microbiome Group. These are people that, as we sit and attend meetings like this, they're actually in the lab generating the data and also working on different projects. I could spend probably a whole day trying to talk you through all of the different projects that we have in the lab. You know, we've got people that work from everything from poop to air quality in a number of different settings. And a few of the postdocs that are in the group here will start to maybe give you an overview of the projects that they are leading within our research program, and also try to give you a sense of the societal impact that these projects potentially have. So with that, then I'll ask Nello to come and give the first presentation, where he talks about his experience and also background.
Thank you. Hello, everyone. I'm Nello. So my talk to you guys will be a really quick thing about my experience. So this is my first postdoc since finishing my PhD over in the UK, and then kind of what my project that I'm developing with the African Microbiome Group is, and kind of how you guys may fit into that. So I did my PhD at the University of Warwick, and basically I looked at ocean algae and bacteria interactions using lab cultures. So I did my focus was on Micromonas, where I looked at the production of DMSP, dimethyl sulfonyl propionate, which Mai will also talk a little bit about as well. It's really important for biogeochemical cycling and the sulfur cycle specifically. This utilized a bunch of culturing, LCMS, GCMS, and FPD detectors. So I have a quite good understanding of how to grow stuff, as well as analytical techniques. I then did amplicon sequencing to see kind of what else was in these cultures, because they're unique algal, but not xenic. And then I did whole genome sequencing of these isolates that I was able to gain to kind of start putting together how these organisms work together to cycle sulfur in this case, but also what these bacteria give back to the algae in trade. But today I'm going to talk about what I'm doing now. And so obviously I work predominantly with model organisms, and now it's kind of more in situ measurements. So as all of us know, coastal regions are super important. It is the only environment that I know of that incorporates marine, freshwater, soil, sediment, and also air microbiomes, and also environments. Also, as most of you know, it's kind of really suffering under climate change as well as anthropogenic sources. And this is predominantly due to marine pollution, which originates from terrestrials such as ourselves. And then this basically slowly comes through the freshwater system, also through the air into the marine system. This is causing quite a lot of bad health effects towards aquatic and terrestrial organisms. And basically that means we're seeing a reduction in soil health, in water quality, increased risks of illness for aquatic organisms as well as humans ourselves. So this also really feeds into the One Health kind of framework. Work has predominantly only focused on how microorganisms biodegrade these compounds or these emerging contaminants. Not a lot of work has actually been done to show what happens to the ecosystem services within this super important environment. Like I surf, I like scuba dive as well, and like you can see the amount of plastics, the amount of rubbish. Also, I know quite a lot of people get ill when they go surfing here, which is, yeah, sad. But luckily there's enough pharmaceuticals in the water to kind of balance that. But yeah, so at the moment I'm running or designing a pilot study in False Bay. And the reason being is that False Bay had a bunch of wastewater treatment plants, which all feed into the bay. That might be fine, but due to the oceanography and geology of it, what you see is with the currents is that it forces a lot of the debris into the Macassar Strand Gordons Bay area, and it basically is trapped there and slowly deposits. The reason why this is bad is a lot of pharmaceuticals, synthetic as well as naturally occurring compounds, then slowly accumulate in the sediment, in the soil, in the aquatic life, and also in the seawater. I've just used, so salicylic acid is also a phytohormone. It's used in a lot of cosmetics. And so we don't actually know how this is going to affect such a large increase in a phytohormone onto the microbial community. Really sadly, coastal water quality is only checked in one way, and that is through fecal bioindicators, so E. coli, for example, or enterococci. You are missing loads of other information just looking at that. So you don't really know what is happening to the rest of the community or animals there. So this is kind of the main aims of this small project study, is to just establish microbial monitoring within the bay, and then also to investigate specific anthropogenic pollutants within the bay to see how they affect the community. And then finally is to isolate and characterize microorganisms within the bay that might have the ability to degrade these compounds, which can then be used as biotechnological applications for reducing these compounds, either incorporating them into a kind of wastewater treatment plant, or perhaps maybe like further afield. I'll be doing this through a wide range of different methodologies. So I've chosen to really focus along here. So this is obviously Strand, Macassar, and Gordons Bay, which should have like really high concentrations of these compounds. And then as you slowly leave the bay out, you will hopefully see a reduction in these. Really, really important is here, Betty's Bay is the African colony of penguins. There's only 2 in South Africa. One is there, then obviously the other one is on the other side of the bay. And hopefully this is a marine protected area, so we should see pristine. Should is probably the apt word. So I'll be using a bunch of different techniques from isolations, flow cytometry, DNA extractions, and qPCR, and then obviously also using barcode amplicon sequencing to produce, hopefully, a kind of snapshot of water health within it, as well as perhaps potential strategies to mitigate them. And this is obviously will be used through data storage and submission to databases, and also hopefully publication to kind of use this to springboard into the main thing, which is setting up a coastal monitoring points along South Africa's coast. Because obviously this is not only in False Bay, it's also really bad in Durban, I know, and also considering South Africa's coastline is about 18,000 miles. I don't know what that is in kilometers. I haven't twigged back yet. But there's loads of other opportunities to start looking along the coastline to see what is defined as a pristine environment and what is now really like heavily like toxic in some ways. But thank you. I'm going to pass this on to Sama. Thank you very much.
Hi, everyone. My name is Zalma. I am also a postdoc in the African Microbiome Group. I I did my PhD and then my postdoc, first year postdoc in the Biomedical Research Institute at Tygerberg in Stellenbosch. So I'm coming from the health sector and then joining this side. So what I did, because I just recently joined, so it's been a month. So what I specialize on was understanding the aerobiology of MTB, which is an airborne pathogen. And then moving from there, what we do actually is to see how long the aerosols survive in the air. And then for that, there was a slide missing here to kind of give that brief introduction. But then what I looked at here was to show you that with patients that we sampled aerosols from, so we were collecting aerosols from when they are just standing and breathing normally. And then the other part was spontaneous cough, when you just cough, normally you're not forcing it or anything. And then the last one was forced cough, when we asked them to cough at least 100 times. And then we looked at those differences if those aerosols contain any culturable bacilli in there. So if it's culturable, we say it is infectious, so that person carries potentially infectious aerosols that could infect the next person who's susceptible. So we looked at how tidal breathing, spontaneous cough, and forced cough differed. And then for this one, we're just looking at the aerosol CFU coliforming units. And then for this one, we had a patient where they wore a face covering, and then the other one, they were not wearing a face covering. So we did it was a tiered study where we also determining if using a face mask, and does it reduce infection of aerosols, and then does transmission in the air. So with tidal breathing, we really had low numbers in this one. So yeah, there was a reduction in all of these, and then we saw that forced cough contained higher aerosols compared to tidal breathing. But then that doesn't mean tidal breathing on its own is not infectious. It's still infectious depending on how long you stay in that room. So that's what we're trying to look at, and just show that breathing is also as infectious as cough. And then the other part was to look at now, so when you breathe and respiration, you generate a different, the diameter of your aerosols, they differ. So we wanted to see which ones carry infectious aerosols. So in that, we saw that, which one is it? Okay, so we saw that the ones that are in stage 3, 4, and 5, I think the range is 3, 4, 5, between 2 microns to 2 to 3 microns, 2 to 4 microns around there. That's when you have a lot of aerosols, and those are the ones that linger in the air and could be infectious. And you can also, so with the black dots there, those are the aerosols containing culturable bacilli when patients were wearing a face covering. And then when they were not wearing a face covering, you can see that it's much higher. And then this was the interest of the study, looking at that the lower respiratory tract, because that's where we find all of these infectious pathogens. So we also looked at particle counts. So those particles, we didn't look whether it's infectious aerosols or not. So we're just looking at the number of the quantifying particles that they generated at that time. So what was interesting for us was to see that tidal breathing actually generated more particle counts than spontaneous cough, but then forced cough still had the highest number of particles. And then we also see in that figure on the other side that where they wore face covering, there was quite there was a significant reduction of particle counts in spontaneous cough and forced cough. And then the other part, and then I'm just going to take this into now the environmental part of looking at the bioaerosols, which is what I'll be focusing on for the duration of my study in the group. So I'm interested in looking at aerosols, the pathogenic bacteria in the air, mostly in areas that wastewater and other water bodies. So with this, I want to see the interaction between pathogenic and non-pathogenic bacteria and see how this may enable the survival of pathogenic bacteria. And this is just showing you how aerosols form in different, okay, this is a diagram in the oceans. So you see that there's a wave breaking, and then you see some microorganisms there that are taken up into the air. Some may be aerosolized, some might not, and then will land back into the oceans. And then you also have other communities that are in the ocean. So for me, it would be nice to understand how does it differ, the community of those microbes, how do they differ when they move from the water and then into the air, and then they're dispersed all around and for transmission. So what biomarkers are there that could enable us to identify pathogens at an early stage? So with this, I want to look at using culture dependent and culture independent methods. And then with the sample using the dry air method. So with that one, it's called a corrosive impact. What it does is just sample aerosols for, it can go for up to 8 hours, I think. And then from that, you can use the liquid, I think it's 15 mils, and then you can use that to either extract DNA or culture it. And then the other one is to use, I can't remember the name of that one. I think Nancy also uses that. So with that, I want to use gelatin filters to sample aerosols. So they'll be on, and then collect them from there. What I like about gelatin filters that it's they might be able to retain more DNA. With experience, we used it when we were collecting MTB, Microbacterium tuberculosis bacilli. So we use a mask, and then we have gelatin filters inserted inside the mask, so that we use those we remove those gelatin filters. It could either be as a strip on the side, or it was in front of the mask, so that when they breathe, we capture those and then dissolve it and then take the gelatin filter, dissolve it, and then culture or do DNA extraction. And then the second method is to use the ACI, so it's an Anderson cascade impactor. What it has is it has pores inside the plate. It's a metal plate, so you can extract the different diameters of aerosols in that, and you can see which range, which is what I use to see at what size range are the infectious aerosols in. And then from that, I will culture and then try to isolate to get single isolates, and then for pure cultures, and then further use that for DNA extraction or to identify the bacteria that are found in there. So this is more of like an in situ sampling. And then with that understanding, the plan is to now use that community of microbes that we find in the environments, and then simulate that into understanding how do they survive within a chamber. So with this one, it was still the aerosolization of mycobacteria. So I'll aerosolize mycobacteria from the side, and then it stays in the chamber, and then collect that into solid culture. So it has 6 stages, which I showed in the diagram previously. And then so for solid culture and liquid culture, because we've seen that solid culture and liquid culture, they can microbes can react differently depending on the culture method. And then so yeah, so this is just a setup and then cleaning of this of the whole experimental setup. So it will be nice to move from understanding how they live in the environment, and then into and then studying that and manipulate certain conditions to understand the pathogen survival and ultimately the human health. So I think I'll end it there for now. That will be my presentation. Thank you.
Greetings, everyone. Thank you so much for this opportunity. But before starting with this topic, I would like you to imagine growing up in an environment where the air you breathe is silently affecting your lungs, but not only your lungs, but also your brain and your gut. It's not a good imagination. But this is a reality for many people living in Saldanha Bay, where the air is filled with particulate matter known as PM, which carries toxic metals like arsenic and lead. And also they carry invisible microbes such as viruses, bacteria, and fungi, which are collectively known as airborne microbiome. So today I'll just share our research on how we are investigating the health impact of airborne microbiomes and toxic metals in people living in Saldanha Bay. So just to give you a brief overview of what Saldanha Bay look like. So it's a coastal industrial hub. There's so many people living there, but there's also a lot of industrial activities happening. So there's a major port receiving minerals from Northern Cape, mineral ore from most of the mines located in Northern Cape. So they also process these minerals, and there is also marine aerosols and bioaerosols, biomass burning. So all of these activities, they produce this particulate matter, which paints the city dark. So they call it red dust. So you see it everywhere. Their homes, dogs, you see it with this color on their bed, like everywhere. So they are really concerned. But one of the things they are not aware of is that these dust that are visible, they have the capacity to carry toxic metals that when you breathe, they can affect your lungs. They can also lead to cancer, and they also carry pathogenic microorganisms. So as a group, before showing you how we are trying to understand the health impact that the red dust is doing, I just want to give you a brief of what's in the air in general, not only in Saldanha Bay. So we have these dust particles known as particulate matter, and these can be inhaled, especially the 2.5 microns and also 10 microns. They can be inhaled by any human being or living organisms. But they carry biological components, inorganic components, and organic components. And some of them is viruses, such as de novo virus, aspergillus, fungi, and legionellosis. They also can be carried by these particles. And they act as a transport mechanism, so they have the ability to transport them everywhere. So what happens? So what happens when we breathe this particulate matter containing these toxic metals? They travel from the lungs to the gut directly, either by swallowing or through the gut-lung axis. And once they are in our system, it can lead to cognitive impairments or immune dysfunction or long-term problems such as malnutrition and stunting, especially in children. So our aim to try and understand this and come up with solutions. So we want to evaluate the potential health risk by integrating environmental sampling with microbial and geochemical analysis. So what I did, so currently we don't have results, but we have already collected the first batch of samples. So I went to Saldanha Bay for a week. This is an image example of our equipment. It's called SAS 3100. So it's a high volume air filter. So it's operated 300 liters per minute. So it sucks the air, and then we're using a filter. So we have two filters, one collecting the biological material and the other collecting chemical analysis. So during this sampling, I have to wear this suit just to avoid contaminating the samples with my own DNA. It's very hot, but yeah, it looks cool. So this is one of the areas we collected from different residential areas, industrial zones, and also coastal areas, just to see. So this is one of the coastal areas. There's a BNB next to it. So this will, I'm hoping, I'm hypothesizing that it will show us if these the influence of the sea is transporting this particulate matter to the residential areas. And this is the industrial zone, and this is one of the residential areas. So the filters were interesting. So we put it up for 6 hours, and we did it 3 times. So went back 3 times to the same site, just to make sure that we get good statistical power. And these are some of the filters that we got from that. You can visibly see that this industrial, I think this is the IDZ industrial zone. It's very contaminated, and this is the this is the residential. You can see different colors as well, even in the filters. So it will really be great. So I can't wait to see the results, both of the geochemistry analysis and also the microbial. And one of the things I read is that arsenic acid also leads to resistance to antibiotics. So with us looking at both geochemistry and also microbial, it will help us understand more about what's happening. So after this, doing DNA extraction and RNA isolation, so that I can, and I'm also doing qPCR just to quantify the different pathogens that can possibly be there, and this will help us more of calculating the microbial risk, comparing it to the WHO standards, and see whether, comparing adults and also children, what risk they're exposed to. And then we're also doing short-term metagenomics to just see the overall microbial community, known and unknown microbial community, bacteria, viruses, and fungal. And with the second filter, it goes through digestion and geochemistry analysis using the ICP-MS. And this will also give us more of the chemical risk assessment. So we'll be able to quantify the different metals and chemicals, and also try and calculate the risk. So with this study, it will help us map the microbial and chemical exposure risk, highlighting the high risk areas, especially in Saldanha, but it's also applicable in any other areas, because we are also doing this study in informal settlements. We're trying to collect an informal settlement and see if it correlates with the gut microbiome of children living there. So this is another different part of the study. So this study is, it will encourage the One Health policy, because it perfectly links the environmental and policy health outcomes. And this will also offer a basis for targeted intervention to reduce the risk in under-resourced community, developing new microbiome-based methods, and also just to give more awareness to the community about what's happening. So with my conclusion, so this study is, I would say it prioritizes transdisciplinary research to break the cycle of environmental gut dysfunction. So it's not only one field that can solve this problem, but working together with the community, policymakers, and microbiologists can help us move closer to the solutions. So with that, thank you so much.
Good morning, everyone. My name is Maibongo Buthelezi. And today I'm going to talk to you about microbial dimethyl sulfonylpropionate, which Nelo nicely introduced in his talk. I'll be referring to it as well. I'll be referring to it as DMSP throughout my talk. I hope to shed light on its ecological and physiological importance on marine microorganisms. So the basis of this study is actually to try understand mechanism that microorganisms, is to actually try to understand the mechanism that microorganisms they are engaging during the environmental fluctuations in a manner that does not interfere with their normal metabolic activity. And really, this compound DMSP fits very well because it's very important to support microorganisms during environmental changes. So it's one of the most abundant organic sulfur compound in the marine environment, and it accounts for approximately 15% of microbial carbon that they fix within the marine environment. So it's actually both bacteria and algae that synthesize DMSP for its anti-stress role to protect themselves against environmental changes such as changes in temperature, salinity, nutrients availability, oxidation, and pressure. But also microorganisms, they degrade DMSP to derive their requirements for organic sulfur and carbon. But it is through the degradation of these compounds that these two, can I point with this?
Mm-hmm.
Well,
It moves your hand.
But yes, it is through the degradation of this compound that these two compounds, dimethyl sulfide and which I would call it DMS, and methanethiol, which is mesh, they are produced very volatile organic sulfur compound that sometimes birds they use their smells to detect some of the productive regions within the marine environment for foraging. It is very important also for climate cooling because of it. They have what they call albedo effect, which helps in the formation of clouds. So with that being said, there's still a huge research gap in very important marine environments, such as the Atlantic Ocean. Across the entire Atlantic Ocean, I refer to it as AMT, which is Atlantic Meridional Transit. This is the study we conducted in 2023, where we collected samples across the Atlantic Ocean. And basically, we formulated this basic question, is DMSP biogeographical traits being influenced by environmental parameters? Do microbes express functions related to DMSP production and its degradation? What are the key microbial drivers of DMSP across the AMT? And this is just our basic workflow from sample collection, collecting samples for DMSP measurements, and synthesizing our metagenomics and metatranscriptomics data to look at the presence and the activity of these genes that are involved in the production and the degradation of this compound. So what did our results show us? So we first saw, so these, the line plots are DMSP concentration and the bar plots are cell abundance or microbial abundance. So we first saw that DMSP is detectable in, number one, in all the samples, but we saw like a variation between oceanographic species of the Atlantic Ocean, and they're tightly correlated with the cell abundance across the samples. So that's number one, tells us that the concentration of DMSP, that number one, they are dependent on microbial abundance within the Atlantic Ocean. But also now we try to correlate DMSP or to gauge the relationship between DMSP and many environmental parameters such as temperature, salinity, including cell abundance, pH as well. We saw like a nice clustering or grouping together of samples that were collected from the same region, particularly within the South Atlantic gyre of the Atlantic, within the South Atlantic gyre of the Atlantic Ocean. So this already shows us that there is about geochemical traits of DMSP that are driven by environmental parameters within this environment. But we further advance with our analysis to try and understand microbial genes, which are responsible for the production and the degradation of DMSP. So, for example, these DSYPs or these 1, 2 here, they are producers, and these ones, ALMA 1, 2, this DMDA, they are degraders, and also we identify the ancillary genes that helps microorganism to actually recognize DMSP. Those that cannot synthesize DMSP by themselves, so they will have to first recognize it from the environment, transport it into the cell, and also helps in the upregulation of metabolizing DMSP when it's within the cells. And that's exactly what we see, that microorganisms, they express them. If you see like the presence of this gene, it's in low abundance, but its expression is very high. This gene is for DMSP transportation, which shows that those organisms, even those organisms that maybe they don't have capacity within themselves to synthesize DMSP, but they have a strong need for this compound, which they import it into their cell and start to metabolize it. So, and then obviously we analyze our data in different layers. So this is still in context level, and we're trying to look at the key organisms that actually contain, that actually express this RNA data, that actually express DMSP within this environment. I'm not going to go through each of these microorganisms then, but we show that both bacteria and algae, they, in their diversity, they express or they show the need within the Atlantic environment of DMSP. Probably they are synthesizing it to counter the environmental changes that we observed on the previous slide, but somewhat this is the limitation of the study, because we can form on our observation, we can synthesize the knowledge, but obviously there will be those limitations that we have to specifically investigate that parameter and why organisms respond to produce DMSP in those environmental conditions. So now we synthesized high quality metagenomics assembled genomes. So this is just to look at the genome level and on each microbial genomes that we reconstructed, if they have any functions that are related to DMSP. So we generated 81 high quality mags. They grouped into 19 order taxonomic rank. And we first saw that some of them contain DMSP producing genes, but majority of them, which are these genes, they contain these genes that are important for DMSP degradation, which shows the importance of these compounds for these microorganisms within the Atlantic Ocean. So what we did again, we tried to map our genomes to RNA sequences to try and see, just to gauge the activity of these mags by gaining their mapping with RNA sequences, and also to get their distribution across the Atlantic Ocean. Yes, so I forgot to mention like that we also investigated the genome sizes of these genomes. They range between 1 million base pairs up to 7 million base pairs, showing like the diversity of microorganisms or the metabolic complexity within these microorganisms. So in our study, we conclude that DMSP concentration exhibited different relationship between abiotic and biotic factors across the Atlantic Ocean. And within this environment, microorganisms contained and express genes encoding for DMSP production and degradation. And it was interesting to see that it was actually, it's just that that data is not included here, but it was interesting to see that those microorganisms that were expressing the need or expressing genes that are encoding for DMSP production were those that were occurring in higher abundance within the Atlantic Ocean. And obviously, these results can be attributed that microorganisms, they show a need for this compound to counter the environmental fluctuations within the Atlantic Ocean. So with that being said, the research which I'm proposing to do now across the South African coastal ecosystem is to number one, to investigate the role of microbial DMSP across along the South African coastal ecosystems with focused research questions. Can the relationship between microbial DMSP and environmental factors be a useful indicator for changes in the environment? What are the key microorganisms that drive these DMSP processes, and why microbial taxa are triggered to engage this pathway, if that's the case, along the South African coastal ecosystem? And we want to know, does DMSP-related pathways contribute to climate regulation along our coast? Because I think it's one of the knowledge gap that we don't have, if these compounds that are produced to also contribute in climate regulation along our coast. And we hypothesize that coastal ecosystem may have high levels of DMSP contributing to global cycling and climate regulation. And we predict that because there hasn't been an attempt that I know of to look at these, that high proportion of novel microbial taxa may drive DMSP production processes in this ecosystem, given like that the strains that you know that has been shown to produce this compound in the laboratory, they are all derived from the other part of the world in the Northern Hemisphere. With our biodiversity and the differences in this ecosystem, it's probably possible that these processes are driven by different microbial organisms or maybe diverse microbial types. I aim to do this research in marine water, estuaries, and sediments. The coastal region also has been shown like to be basically these ecosystem, particularly estuaries and sediments, to have to show a very enhanced process, a very enhanced DMSP related processes that you see from the literature from on the other part of the world. And my object, this is the methodology and sample processing for my objective one, basically collecting those samples and measuring these compounds, generating data for genomics sequencing to actually get the mechanistic side of inside of these microorganisms to conduct these processes. I aim to venture into doing a stable isotope probe, where we'll be trying to incorporate the heavy chains of DMSP to see which actually taxa are metabolizing DMSP and for what specific reason are these taxa metabolizing DMSP. And like almost everyone in the group, we're gonna try to also culture some microbes to actually get the novelty, which I hypothesize that probably there are some microorganisms that novel or previously undescribed microorganism or genes that are involved in these processes across our coastal regions. And I think this is my last slide.
Good afternoon, everyone. Thank you for having me here. Yeah. So my name is Sam Leering, and I'm also part of the group. And I will be talking to you about, I think you've heard about air microbiomes and marine microbiomes. And now we'll be specifically focusing on freshwater and wastewater, which I think a lot of people had a lot to say earlier, so it has saved me a lot I had a background which just basically talks about how aquatic systems are facing anthropogenic inputs that are affecting them. They're affecting both the water quality and of course microorganisms that are driving all biogeochemical cycling processes as well as ecosystem services. And I have this nice figure here, which is for a purpose to make you really look hard at it and realize the impact of these inputs on aquatic systems. And a lot of these inputs include emerging contaminants, excess nutrients, antimicrobial resistance genes, as well as microorganisms, some of them which are antimicrobial resistant. Seem to be struggling with this. Just use the arrows as well. Fine.
We have a problem.
But maybe you can tell us what you.
Yes.
Because we also want to know.
Yes. So this is just an overview of the pressures that freshwater systems are facing, especially rivers, and those that are specifically associated with wastewater treatment plants. I had a nice diagram there, which I was going to talk you through.
We can imagine.
Yes, to tell you the importance of why we need to look at specifically wastewater treatment plants and their context within the One Health concept. And from there, we see that these are the chemicals that are released into this wastewater treatment plants from different sources, such as our households, which of course links to our activities, as well as other non-point sources, such as agricultural runoffs, end up in the systems. And in South Africa specifically, as well as regionally, we have the challenge where our wastewater treatment plants are often over capacity, and they do not do a good job of removing some of these pollutants that end up there. And in turn, they are released into the environment. And this leads to challenges such as eutrophication, which affects our water quality, and also affects microbial organisms, as I said. So we then took it upon ourselves to look at some of the outcomes or consequences of these pollutants in freshwater systems. And I was going to talk to you about some of the projects we are doing. One of them being a local project where we characterize the rhizostome across Pretoria, Tshwane. And we looked at a metagenomic approach to just fully characterize resistance from rivers that receive wastewater. And some of these differ by sizes as well as treatment options or processes and technologies. And so we wanted to get a full idea of what happens once the wastewater is released into the environment and how it modulates these ARGs. And what we found was that compartmentalization usually drives ARG proliferation and dissemination. What that means is that the ARGs, in some cases, their abundances and richness are increased by the wastewater treatment processes due to the selective pressures that are found there because of the chemicals that occur there. Once again, I had a nice visualization, so I'm trying to paint a picture for you to get this.
Doing a great job.
Yes. So yeah, so we saw that compartmentalization was a major driver of these ARG patterns that we observed. So this was, as I said, a regional project, and from what we learned from that, we also wanted to broaden the scope to actually take it to the region and include other collaborators as well, where we would use the same tools and approaches and sort of improve some of the aspects to look at characterizing rhizostomes within wastewater treatment plants and receiving waters across the continent. And so far we have 11 participants or 11 participating countries, and the number is always increasing. I think we have recently had talks with people in Tunisia who also want to join the project. So we will be standardizing the sampling, the sequencing, as well as the micropollutant, antimicrobial load measurements or concentration quantifications, where we could actually look at what actually drives the resistance across these different countries, as well as sort of get a baseline understanding of antimicrobial resistance in Africa. The other project that we have been running as well to just fully understand how antimicrobial resistance forms was where we looked at short-term temporal variations from human activities like wastewater inflows from when we use the bathrooms and how that affects antimicrobial resistance profiles. And from that, we definitely saw variations across times. Funny enough, interestingly, we saw that at midnight was when we saw this increased richness and diversity of antimicrobial resistance genes and wastewater treatment plants, which talks to how we approach sampling for characterization of resistomes. So that is an interesting aspect that we will be considering for sampling for the broader projects that we will be doing in this area. From there, we also have projects that look at how these chemical pollutants that are released from wastewater treatment plants actually affect microbial community structures and functioning, specifically on biogeochemical cycling, and how this could potentially be used as microbial indicators of pollution. So we have a running project that I thought I would demonstrate as well, so that we could all see where we could collaborate, where we intend to look at spatial as well as temporal variations in terms of microbial community structures and functions and how they're affected. We are currently sampling, or we will be sampling in Pretoria, as well as the Western Cape and potentially Durban. And we are always looking for collaborators to join the project and make it more extensive so that we could look at different regions and all these variations, look at potential drivers, ecological drivers of microbial functions and community structure. And I think that is my talk.
What Sam experienced was calmer, because we were supposed to have 10 slides each, less. He had 16. That's why the computer decided to give up. But the whole idea of this, I hope you could, I know it's a lot to pack into a morning, but what we try to demonstrate is the capacity in terms of the skills that we have within the group and the potential for collaboration. You know, as we were saying in the morning, we're trying to move away from having single labs work on a single question and try and create a platform where we can have people with diverse skills work together to tackle some of the common challenges that we have. And I'm interested to see how in the next 3 days, we can all start to explore potential projects that we can collaborate on, and maybe think about how we can exchange students, how we can sort of add on to this framework other skills that we may not have considered. I think, especially in a diminishing funding environment, we all need to start thinking a bit more about how we can start to collaborate. But thanks for your patience and for outlasting the computer. And I hope you enjoyed those talks. So thank you very much.
Okay, thanks, Sam, and the team. High-powered delegation indeed. Yeah, thanks to Thulani and the team, right? Sam, sorry about the technical glitch. You did exceptionally well there. So we'll take a few questions, comments, suggestions. I think there's a lot to process from the work that has been shared by the team. Really exciting. I think the linkages from the environment all the way up to the societal aspects, it's really exceptional. It's too late for me to run away from fish though, so I was trying to see where fish would fit into all that, and I think there are a number of possibilities. But yeah, let's get some questions from the we have a question here, Funza, and yes. We start with you, yes.
Okay, so my question is to Mr. Mayambongwe. For the mapping of the genomes to the RNA sequences, what binning software did you use?
So,
You understand that?
You can come to the front, yeah, for the colleagues online. Yes.
okay. So, thank you for the question. So we use MetaPad 2 for our binning, but we do not use the binning tool for mapping the genomes to reads. So there's another tool that we use called CoverM.
Sorry?
Cover, yeah, CoverM. Yeah, it's CoverM. But it's not only that, you can also use BBMap if you want. Yeah, and there are so many parameters that you can select at what RPKM or whatever average that you want, whatever insight that you want to gain from actually the reflection of your genomes to your RNA.
Thank you. Colleagues, just go ahead and ask your question.
You said you found the highest amount of microbial, I think it's midnights.
Yeah, from what we've seen and in other studies, it looks like water usage during the day as well as in the evening drives these patterns. So that is what we're seeing even in wastewater treatment processes where they receive these large volumes, and the treatment processes, and also sort of the retention time and all proliferates the ARGs that we're seeing. Yeah.
So that's basically why we set up this study. So because we wanted to understand if you wanted to work in wastewater treatment plants and so on, when would be the most ideal time to set up this sample? Because you don't want to do it randomly because you want to have that bit of a structure. So, and we wanted to torture them in an honest student. So we put them there, was it for a week? Where they sampled at 6 a.m., 12 p.m., 6 in the evening, and midnight, consistently over a week. And then the combination of optical analysis and shortcut sequencing to try and determine whether we see those diurnal patterns as well, because that will inform when you're by samples.
So I love your work, all your work. Thank you very much for, you are speaking the language that I understand a bit besides the DNA part. So what I want to understand with you integrating the, I think my question is for you, and also you, with integrating the environmental assessment with also the societal part, when you do this risk assessment, do you actually do assessment on people? Do you work with people? And if so, do you, how difficult it is as an environmental person to acquire ethics when you are to work with that kind of project?
Yeah, so I'll just start and someone can add. So with the gut microbiome and which we are looking at conducted in informal segments, that part needs ethical clearance. Yeah, because you need stool samples from mothers and kids. So yeah, so we are working with another group. So it's a multidisciplinary study. So we are working with economists. So they conduct, so they have workshops, so where you speak to the community, tell them about the study, and then they also give you consent, and then yeah, so that process makes things easier. And then the air samples, it's easier. That's why I enjoy more working with environmental health, because it's more of like you don't get to trouble people. And yeah, so collecting the air sample is nice and easy. So people, when they see me, even in Saldanha Bay, they get to ask questions because they are very concerned about the red dust. So they ask questions and get to interact with them, but they don't have a problem. So that part there is not the problem. So, yeah. I don't know if I answered your question.
No, you did.
Thank you. Before Rosie, there was Pumza and then Rosie.
I have a question for Sam. Sam, will you just focus on the water samples or you also collect in the sediments?
Yeah, we also do sediment sampling.
Oh, and so-
Yeah, so I think because I was just trying to summarize it without bombarding you with a lot of information, I skipped that part. Yeah, but it was nicely represented. Yes. So we are trying to look at incorporating or synergizing both microbial ecology, which is microbial data, as well as looking at toxicology as a measure of looking at the risks of antimicrobial resistance. So that is why we're interested in looking at what is an environment and how it drives this antimicrobial resistance profiles that we are seeing. So rather than just looking at profiling, we are interested in painting a fuller picture of the ecological drivers as well.
Rosie?
I have two questions as follow-up for Sam. The first one is the diurnal patterns that you're seeing. Is that for bacterial growth or is it for expression of the resistance genes?
It is not expression, but it is a coverage of those genes within the metagenomes.
So does that mean that these resistant bacteria are coming from the people that are using the system and not necessarily increases in bacterial growth in the sewage farm?
So some of the hosts, I think some of the hosts that I've found in the results are environmental bacteria. So there is exchange of horizontal gene transfer processes that take place because of plasmids in such an environment, especially in the wastewater treatment plant. But we also have hosts that are of human origin or from the gut. We have a number of taxa that are from the gut and they end up within the wastewater treatment plants, and some of them cannot be effectively removed by wastewater treatment plants.
Yeah, it's interesting that the bacterial numbers are changing. That means that there must be spurts of growth after the sewage has been discharged into the system. And it'd be interesting to know if non-resistant bacteria are also, there's an increase in metabolic activity.
In terms of microbial community structure and composition, there are, of course, there are diurnal influences, smaller variations that we see, but they are not significant and they're stable across those times. So, yeah.
And then I have a question for almost Dr. Buthelezi. My question about DMSP is, I understand that it's produced by phytoplankton. So when you talk about microbial communities, are you talking about microplankton as well?
So,
or are you just talking about bacteria?
That's it.
I would say maybe look at the people. So
I think that has been almost a knowledge on the entire research of DMSP for the entire time of in DMSP research that it's solely produced by phytoplankton. But also bacteria produce it, and people who are doing like the phylogenomics of these genes, they show that actually this gene they first originated with bacteria and or from the bacteria, maybe the horizontal gene transfer or whatever they were acquired by phytoplankton. So there is a very strong evidence. Actually, the first mechanistic insight of showing an organism producing DMSP was from the bacteria. And then later, following that method, they show to phytoplankton.
So are they chloroplast genes?
DMS, I think so, because DMSP is synthesized from the chloroplast as well.
Yeah, so it's localized in chloroplasts and mitochondria. Whether the proteins are there depends a lot on the organism itself. So usually they're cytosolic depending on it, because it's but depending on the bacterial or phytoplankton.
Because a lot of dinoflagellates have microbial communities that are intracellular. So it would be interesting to know whether the taxa that you are identifying are free living, or whether they're actually present in the diatoms and dinoflagellates filter bacteria as well.
So when we do our sampling, we fractionate the samples
Yeah,
And
if you
then
look at your fractions,
targeting.
you might find that some of them are in the 20 micron fraction.
Yeah,
Whereas if they're all or nothing, 0.2 micron fractions will have everything.
yeah. So like the 0.2 micron, which we associated with free living, there's quite a lot of these functions as well, which are present and expressed. So I think they are being also expressed by the free living organisms.
Yeah, it's really interesting. Yeah, thank you.
Thank you. We'll get one last question and then I'll ask Rosie to thank the delegation. And then we can yeah.
My question is for Nancy Kevin. I might have missed it, but I wanted to know once at the end of your study, once you find the airborne toxins, are you going to recommend changes to the industry, or what's the overall impact of your study?
Yeah, so with the Saldanha Bay case, so in the Saldanha Bay case, they haven't done any microbial analysis on the dust. So it will be more of informing them of what's happening, and also after calculating the risk, and if it's above the threshold that's recommended by WHO, obviously we also advise on that. Yeah, so it's more of awareness for now. So obviously the government is in charge of the stopping where and how. Yeah.
Thank you. Rosie, could you do the honors, please? Thank you.
Which way is the? Oh, it moves.
Yeah, yeah.
Good grief. That's kind of scary. Oh, somebody moves it. Thank you very much to our visitors from Stellenbosch, the delegation. In particular, it's not always easy to travel all the way to the Eastern Cape and then navigate your way all the way to Makanda. And I've already warned the delegation to be careful of donkeys and cows on the road. But it's always a pleasure to be able to listen to the work that's being done elsewhere in the country. Often we are so siloed and so focused on our own projects that we often miss the really inspiring work that's being done everywhere else. For me as a microbiologist, the most important thing, take-home message from the work that's being done by the group at Stellenbosch is how important microbial communities are, and that they really form the foundation of all the environmental ecosystems that we work in. And just because we can't see them doesn't mean to say they're not really important. They are really important, and we should not discount the function of working in healthy microbial communities in keeping our ecosystems healthy and being able to support us. One of the problems, and this is why I think the African Microbiome Project is so important, is Albert and I have been doing some work on the immense diversity on the only continent that spans all four hemispheres, northern, southern, eastern, and west. And the fact that we are rapidly losing biodiversity that we don't even know we have. And so it's really important to be able to start, even if it's in silico only, to start archiving evidence of this biodiversity, because once it's gone, it's gone. So thank you all for making the trip up here. And I hope you have a really good time. Please take time to go and look at the coelacanth and the story of the coelacanth still.